All right, next up we've got the cellular level of organization, part two, where we're going to be talking about the activities of the cell within the nucleus, this green booger right here, DNA, which stands for deoxyribonucleic acid, and protein synthesis, how that all occurs here. So, no pressure, no problem. We will cover this from soup to nuts in the most simplest way possible. Here's a nucleus, all right? Then you got to know that the nucleus is at the center of it all, literally and figuratively. It is the control center of the cell. So here we've got the nucleus illuminated in a blue stain of some sort. That's where it all happens. Protein synthesis happens primarily within the structure, or at least from this structure. This is where the control center is. The nucleus contains DNA and enzymes for cellular materials. Something called the nuclear envelope. So this is like our cell membrane. There's a nuclear membrane. Right? So it's happens to be a double membrane with a perinuclear space between the membrane and the nuclear material itself. Some of its features: here's the outer membrane, here's that perinuclear space, the inner membrane, ribosomes, or those protein factories on the outside of the nuclear envelope. So the envelope is going to have those two membranes. It's going to communicate with the cytosol through these pores, and these clamshell looking things, or these little volcanoes, they are, let's try a new color here, that's where stuff comes in and out, all right, through these nuclear pores. So what is within that nucleus? What is in the control room, you ask? All right, there are chromosomes, and that's your DNA molecules plus these proteins that are called histones. There are a couple, at least one, sometimes more nucleoli, or little substructures within the nucleus here, like right there. It's our nucleolus, a little darker area you'll see within the nucleus itself. And there's a matrix within the nucleus, or a nucleoskeleton. So again, there's a little internal framework there consisting of the fluid and the tent poles, and that supports everything. There's also cytosol, or a fluid within that nucleus. What happens in the control room continued? All right. What is the function of the nucleus? It controls the cell by creating proteins. It directs protein synthesis. So it's like the conductor of a symphony. It tells everything what to do and produces all the raw materials that are going to be used out in the cell. So it uses these information called chromosomes. That's basically its owner's manual, its operating manual, are stored in chromosomes, which are comprised of all the things we talked about towards the end of our chemistry lecture. This is the genetic code right here. Remember, complementary base pairs, nucleotides, adenine, thymine, guanine, cytosine pairing up. Well, that all happens. That's all that information is utilized within the nucleus on those genes. Chromosomes are bound to those proteins called histones, and in cells that don't divide, DNA is associated, and associated proteins form what's called chromatin. So if this is the nucleus, this is the cell right here. This is the nucleus. Chromatin is just kind of like loose genetic material all coiled up within the center of that nucleus. The genetic code is also called the triplet code because there's these three nitrogenous bases. Again, here we got adenine, thymine, guanine, cytosine. There's got to be a parent. These three nucleotides or bases are going to code for a protein. There's going to be a complementary coding here. So where there's an adenine, there's going to be thionine, thymine, adenine, guanine, cytosine, and these codes and their mirror images are going to be, you look at this right here, this genetic code. When these instructions start to make proteins, they use a code called a triplet. So each gene that we possess within our cells contains all of the codes that we need to produce a given set of proteins. Deoxyribonucleic acid, DNA, is otherwise known as blank. Deoxyribonucleic acid. DNA controls the protein factory. It dictates the cell structure and function. Gene activation occurs when an enzyme called RNA polymerase, remember enzymes usually often end with ACE, ASE. Gene activation occurs when RNA polymerase binds to the gene first. So that's the first step. In order for a gene to be activated, the enzyme blank functions to start things up. And this is an illustration of RNA polymerase right here. So that steps in, then the gene can be activated. Transcription is defined as the production of ribonucleic acid from deoxyribonucleic acid. So we're taking DNA and we're going to transcribe it, which basically means to copy it, and the copy is going to be termed RNA. There's something called messenger RNA that is going to carry that copy that has been transcribed into the exterior of the nucleus, still within the cell, and it's going to take it within the cytoplasm, and that's where a process called translation occurs. So the steps are the transcription. You take a DNA molecule, you're going to copy it, and then it's called ribonucleic acid. Ribonucleic acid is going to be taken into the cytosol, so outside the nucleus now, by mRNA, which is a messenger RNA. Messenger takes it outside the nucleus. Translation occurs in the cytosol. Translation, basically we're going to take a functional polypeptide and we are able to make it using the information from that messenger RNA or mRNA. And the codon sequences, here you see them, that triplet code. You see that assembling itself, basically the makings of an amino acid right here. These are a protein, rather. You've got alanine, valine, leucine, glycine, methionine. So the sequence of the codon determines the sequence of amino acids. So this little triplet approaches, boom, we're going to make an alanine here. We've got the alanine. Next we're going to move on to a glycine, and you'll see that glycine join the string of beads. And that's what's happening right here. We're creating a protein in the process of translation. tRNA is otherwise known as transfer RNA. This is a general illustration of it. We use the complementary base pairing, triplet code here. tRNA molecules present an anticodon to messenger RNA, bring the amino acids into the ribosomal complex. Our ribosomes are where our proteins are manufactured. So we've got this structure right here. We're creating the complementary base pairs, and that whole illustration we saw previously, boom, up there goes another amino acid on the string of proteins, making a polypeptide. Protein synthesis, all right? The DNA that is situated in the nucleus has both the direct and indirect control over protein synthesis by the means that we just said previously. We've got the elements of transcription, where RNA copy is made from DNA, and then messenger RNA is going to go out here to a ribosome and the cytosol. Translation occurs, and then the protein starts to assume its various folding structures. So something called an alpha helix and a beta pleated sheet. It all starts inside the nucleus, and then the DNA is going to take its influence outside the nucleus, and that's when the proteins start folding up. And that was it. Okay, that's actually a pretty short lecture on a very complex topic. We're going to review it significantly, cover it significantly in the second half of class tomorrow. And so we'll make sure that you are well informed on it. I'll also post some interesting videos on the process that will help you put it all together. Okay? I'll see you tomorrow.